In this video lecture, I will be talking on pre-transplant testing. Prior to graft or organ transplantation, it is essential to make sure that the organ would not be rejected by the recipient's body. Accordingly, tests should be performed to avoid rejection and complication. So before any tissue transplant, some tests should be performed to avoid this rejection, and these tests include: the first test should be done that the donor and recipient blood group should be more or less similar. So ABO antigen compatibility should be tested first before other tests. Since differences in blood group are responsible for most intense graft rejection reactions. The blood group reactions, or the blood group antigens, are expressed on red blood cells, epithelial cells, and endothelial cells. So antibodies produced in the recipient to any of these antigens that are present on transplanted tissue will induce antibody-mediated complement lysis of the incompatible donor cells. So also, tissue typing is very important as a pre-transplant test, and this for typing HLA, to test for HLA compatibility. And as you know that HLA class 1, which is HLA-A, these loci, the three loci, HLA-A, HLA-B, HLA-C, and the three loci within HLA class 2 are HLA-DR, HLA-DQ, HLA-DB. And this is achieved by polymerase chain reaction, or PCR. It is actually, this is very useful for detection of HLA compatibility between donor and recipient, and it is useful to avoid an early rejection, especially the genes within HLA-A, HLA-B, and HLA-DR. Also, another test is very important to be run prior to transplantation. It is enzyme-linked immunosorbent assay, or ELISA, and this helps in avoiding rejection by antibodies. Some individuals may upregulate antibodies to HLA following blood transfusion, or in case of normal physiological process pregnancy, that pregnant lady may have offsprings with different HLA, so each time she mounts antibody to different HLA from her HLA. And the most dangerous antibodies that may lead to rejection are anti-HLA IgG antibodies. So ELISA detects antibodies against genes within HLA-C loci and genes within HLA-DQ and HLA-DB. And also a test known as mixed lymphocyte reaction, or MLR, and this is for lymphocyte cross-match, and it is useful actually for detection class 2 mismatch. However, other tests should be also performed prior or pre-transplantation, and these tests for detection of infection For example, with cytomegalovirus, Epstein-Barr virus, herpes simplex virus, hepatitis B virus, hepatitis C virus, West Nile virus, and tuberculosis. Thank you very much, Professor Ahmed Bolan.